All right, to start this vodcast, we're going to continue looking at the structure of glucose in a bit more detail. And if you look right down the middle of this picture, you can see again the chain form of glucose with the aldehyde group up here at the top, and you get then that 1, 2, 3, 4, 5, and 6 carbon chain. But when it goes into a solution of water, let me play this quick animation. Here comes the chain of glucose. It goes into water, and when it goes into a solution of water, the glucose will fold up and take on that ring form. And you can see it bouncing back and forth right now between two different kinds of glucose, an alpha form and a beta form. When we get back here onto our slide, you can see on the left-hand side we have the alpha configuration. So it says alpha here, and then over on this side we have the beta configuration. And the big difference between the two configurations is the hydroxyl group on the number 1 carbon does vary ever so slightly in its position based on some bond angle stuff. If you have an alpha configuration for glucose, the hydroxyl on the number 1 carbon is in it's called the down, that's a D-O-W-N, down position. But if you get the hydroxyl on the number 1 carbon to be in the up position, that is considered to be the beta configuration. And really the alpha and beta just means this or that, type 1 or type 2, alpha and beta, these two different configurations of glucose. And so we might come across these two different configurations at different times this year as we're looking at some bonding patterns as well. Continuing on then in structures of carbohydrates, we then move up to the disaccharide. So mono, we said, was a single ring of sugar. Di means two, similarly to bi, and the only difference here now is that disaccharides, two sugars, is we're going to have one ring of sugar connecting up to with some sort of a bond now to a second ring of sugar. This now. First one that we're going to take a look at is called maltose, and maltose you may be familiar with is the type of sugar commonly found in, you can see here, a malt beverage has maltose sugar, whereas a shake has a different kind of sugar in it. Malted beverages like beers are oftentimes using a maltose as their sugar in the brewing as well, or malted milk balls. Lots of different malt-based products that use maltose as their sugar. And maltose is really nothing more than two glucose monomers connected together with what is called an alpha-1,4 glycosidic linkage. Now that might sound like a daunting task to understand what alpha-1,4 glycosidic linkage would mean, but let me show you again another quick animation. If I just kind of mouse over this here, I can get those two to go together, and what you can see, let's see if we can hold it in place, the carbon number 1 on this very first glucose was where the bond was formed, and it connects up to the carbon number 4 on the second glucose. And so the hydroxyl group on each side is what's interacting. We kind of were talking in class a little bit about taking the water out to put something together, and so you can see when it goes back together here again. Okay, there we go. You take the water out, in this case you're putting it together, and it's being held together with this single oxygen that is left behind, linking now these two glucose monomers together, and that is called a glycosidic linkage. You can see it in the picture down below. They've even highlighted for us. The hydroxyl group on one side interacts with the hydroxyl group on the other side, but just the hydrogen from this side and the OH from that side will be taken out in the form of water and then be held together in the form of a glycosidic linkage. So that would be dehydration synthesis or a condensation synthesis reaction. You of course can put the water back in to break something apart, and that would be the hydrolysis reaction. So you get the arrows going in each direction. If I'm going to put it together, going this way. If I'm going to break it apart, going that way. In each case, water is involved. Either take the water out to put something together, or put the water in to take it apart. The next type of disaccharide I want to talk about is sucrose. Sucrose, commonly thought of as table sugar, is when you take a glucose monomer and a fructose monomer and you hold it together with an alpha-1,2 glycosidic linkage. Again, it has to do, you can see in the picture here, has to do with which carbon is involved in the bond. The first carbon on that glucose is number 1, and the carbon on the fructose ends up being the number 2 carbon as you count off on that molecule. And that's held together then again with the oxygen in the middle, this glycosidic linkage. I've got two animations here. The second one actually goes into quite a bit more detail, so I will put that out there as a reference for you with the link on Moodle. But this first one will just quickly show us.
A condensation reaction joins two molecules together while releasing a molecule of water.
Through that animation, you can just see the very simple form of glucose and fructose coming together. Again, the second one that I'll put on Moodle gets into quite a bit more detail about exactly how it lines up, exactly how the water is taken out, and just digs a little bit deeper into that. But I'll let you investigate that a little further at your own leisure. Lastly, lactose, another OS sugar, is considered to be a disaccharide because it's a glucose monomer and a galactose monomer that come together with a beta-1,4 glycosidic linkage. And so this one is when the hydroxyl group on the number 1 carbon of that first glucose is in the up position, so it's in the beta. And so you can see up here there was the kind of the down angle, and over here we've kind of got the up angle to show us that linkage ends up being formed with the hydroxyl on that number 1 carbon in the up position. And so lactose commonly found in milk, and you can see we even have some lactose-free milk because some people are lactose intolerant and unable to properly digest and break apart or hydrolyze that beta-1,4 glycosidic linkage. They lack the enzyme to make that happen, and so those people are considered to be lactose intolerant. Continuing on, we've also got the last group that I want to talk about, and that is the polysaccharides. They're poly, meaning many, in this case saccharide sugar units, many sugar monomers connected together with glycosidic linkages, each time an enzyme coming in, taking the water out to put something together through the process of dehydration synthesis. And a couple different polysaccharides I want to talk about. Starch is one that probably many of us know. Starch is how plants store energy. Plants will store starch in their leaves, in their stems, but probably most commonly in their roots, and that's where we tend to dig for the starch. As you think about a potato, that's just a growth off the root of the potato plant, and it's just extra plant storage for their energy, so that they don't waste but store up any extra that they have. Two different types of starches. Amylose is one type of starch, and there's this description here that again seems a little challenging, but what I want to show you is there are different kinds of starches. Amylose is an unbranching chain of glucose. It's purely glucose connected together with glycosidic linkages, one after another after another. It does form this helical pattern. I'll show you what that looks like in just a moment. All of the bonds are alpha-1,4 bonds, one after another, and an amylose starch molecule might be up to 300 glucose monomers in size. And so here's a quick picture of it, and what I want you to picture here is kind of like stretching out a slinky, and you have this helical spiral shape of one glucose after another, so that each of the bonds between each of these glucoses causes the next one to just angle a little bit, so that when you get a big long chain, it forms this spiral shape. And so it's one big long spiral, hundreds of glucose monomers after another, that we can then, and plants can use as a way to store up their energy. A slightly different version of starch is called amylopectin. The difference is amylose was non-branching or unbranching. Amylopectin is highly branched. It is again helical with alpha-1,4 glycosidic linkages. That's what causes it to spiral and make that helices. But about every 25 monomers, you get an alpha-1,6 glycosidic linkage. Down below in the picture here, you get alpha-1,4, alpha-1,4, alpha-1,4. But this is coming off of the 6th carbon at the top of the glucose, and so that ends up being an alpha-1,6 glycosidic linkage, and it causes a branch to form. So here's a chain with a branch, and then that chains on and on with alpha 1,4s that'll start to form a helix. And so it might end up looking a little something like this. What they struggle to do in this picture is show us that it is helical time after time, but you get a bunch of alpha 1,4s, and then boom, these alpha 1,6 branching points that they're pointing out here cause those branches. The amylopectin starch can get quite a bit larger in size, into the thousands of glucose monomers rather than the hundreds. Next, glycogen. Glycogen is how animals store energy. We do not take the glucose and store it in the form of starch. We store our excess glucose first in the form of glycogen. And about 10% of our liver is actually in the form of glycogen, along with about 1% of our muscle. This is where we go to our energy first when we're running out of the sugars in our body. We go to the glycogen and say, whoa, I'm using up a lot of glucose. I need more. Where do I have some? Muscles and liver. I can start breaking down some of this glycogen. So first we have to store it up in the form of glycogen. Again, it's alpha 1,4s and alpha 1,6s. It's more frequent alpha 1,6s, so it's more frequently branched. And over here I just got a snapshot of some branches. You can just see highly branched. It's more densely packed together. You get a lot more glucose densely packed together in these humongous molecules. But it does still, in this picture, they were able to put a little bit of that helicy to it. You can see how it's helical at times with the alpha 1,4s and branching. Oops, I'm sorry. And branching at times right here, I think you might be able to see in red, is a alpha 1,6 glycosidic linkage. And so glycogen is the equivalent to starch. It's the same molecule, just more highly branched as starch in plants. Last couple. These two completely relate to structure rather than energy storage. Cellulose is a commonly used structural carbohydrate in plants. It is unbranched, it is not helical, and it forms bundles of parallel chains. The reason for this has to do with the type of linkage it is. It is insoluble in water. It is still glucose to glucose, but instead of an alpha 1,4 or an alpha 1,6 glycosidic linkage, it is a beta 1,4 linkage. And so if we looked at the picture, and you can see here it's found in the cell wall of plant cells. It makes these sheet-like fibers here, and the cellulose fibril is now broken down into smaller microfibrils. And if we looked at it, it would be like sheets of paper lying on top of each other, where it's just a sheet of glucose and another sheet of glucose. But it's much more linear. It's not helical because of the beta 1,4 glycosidic linkage. Now the key thing that you'll see here is that there is a lot of then hydrogen bonding here and here and here and here and here and here and here. As these sheets lie on top of each other, they are held in place together with hydrogen bonds from one glucose chain to the next, making up this more complex cellulose structure in the cell wall. Now the interesting thing is that very few organisms have the enzymes to hydrolyze a beta 1,4 linkage. We do not have the ability to hydrolyze or to do hydrolysis to that bond. Even the termites that you might remember studying in a freshman biology class, playing around with them, they, yes, can go out and eat plant material and break down the wood, but they need to have the certain types of protists and bacteria inside their intestines to actually break down or hydrolyze that beta 1,4 linkage. The scary thing is that half of the organic carbon on Earth is found in this form of cellulose, and we as humans have no way of breaking this down. In animals, we have a similar structural component. You can see a picture down here below of something called chitin. Yes, that is pronounced chitin, C-H-I-T-I-N. It is found in animals. In particular, it's found in the exoskeletons of arthropods. You might know some of the arthropods as the insects. They have that crunchy outer skeleton or exoskeleton. But it's also found in the cell walls of the fungi kingdom. And this is one of the distinguishing factors between plants and fungi, is something that they have in terms of structural cell walls, a slight variation in terms of that structure.